Good morning. It's September 11th, and this is your daily brief in science. Here's everything you need to know. Recent research presented at the European Association for the Study of Diabetes meeting in Madrid reveals promising results for liraglutide, known commercially as Saxenda, in treating childhood obesity. The study found that children aged 6 to 12 taking liraglutide experienced a 5.8% reduction in body mass index, while those on a placebo saw a 1.6% increase in BM1. Currently, liraglutide is not FDA approved for children under 12, although it has been available for teenagers since 2020 and for adults since 2014. Lead author Professor Claudia Fox emphasized the critical need to address childhood obesity, a chronic condition linked to serious health issues in adulthood. Some medical professionals support the use of medications like liraglutide as obesity rates rise and families struggle to make lifestyle changes. However, experts caution about the potential risks, particularly due to limited data on long-term effects in young children. The study involved 82 participants, averaging 10 years old and starting with a mean weight of approximately 155 pounds, with over half having obesity-related health issues. Participants taking liraglutide reported a higher incidence of side effects, particularly gastrointestinal problems, with 12% experiencing serious adverse events. Novo Nordisk plans to seek FDA approval for liraglutide for younger children, aiming to address the significant gap in treatment options for pediatric obesity. Professor Fox highlighted the potential of liraglutide to improve the lives of children struggling with obesity, a group that currently lacks effective treatment options. The findings underscore the necessity for early intervention in treating childhood obesity to prevent future health complications. The results of this significant trial were published in the New England Journal of Medicine, marking a pivotal moment in the search for effective obesity treatments for children. BridgeBio Pharma has announced the top-line results from its Phase 1/2 adventure study of BBP-631, a gene therapy aimed at treating congenital adrenal hyperplasia, or CAH. Despite demonstrating some efficacy, the company has decided to discontinue the development of BBP-631 for CAH, shifting focus towards gene therapies for diseases with more significant unmet needs. As part of this strategic pivot, BridgeBio plans to reduce its gene therapy budget by over $50 million. The company is also actively seeking partnerships to support the future development of BBP-631 or next-generation gene therapies for CAH, which affects approximately 75,000 individuals in the US and EU due to a deficiency in the 21-hydroxylase enzyme, essential for hormone production. The Phase 1/2 study revealed that all patients in the higher dose cohorts of BBP-631 exhibited increased endogenous cortisol production, marking a notable milestone for CAH treatment. Additionally, the results indicated substantial increases in 11-deoxycortisol and significant reductions in 17-hydroxyprogesterone, suggesting durable activity of the BBP-631 transgene. The therapy was well tolerated, with only mild to moderate treatment-emergent adverse events reported and no serious adverse events noted. In a related development, BridgeBio has received Regenerative Medicine Advanced Therapy designation from the FDA for BBP-812, a gene therapy targeting Canavan disease. Canavan disease affects around 1,000 children in the US and EU, leading to severe developmental challenges and currently lacks approved treatments. If approved, BBP-812 could provide the first therapeutic option for children suffering from Canavan disease, addressing significant unmet medical needs. BBP-812 has also received orphan drug, rare pediatric disease, and fast-track designations from the FDA, which may facilitate its review and approval process. BridgeBio's CEO expressed gratitude to participating families and emphasized the urgency of bringing effective treatments to those affected by rare genetic disorders. Metabolic dysfunction-associated steatotic liver disorder, or MASLD, is increasingly becoming a major concern in public health. The global prevalence of MASLD has surged dramatically from 22% in 1991 to 37% in 2019, with projections suggesting that it could reach 46.1% in males and 41.0% in females by 2030. In Southeast Asia, particularly in the Republic of Korea, the prevalence is notably high, reported at 32.87%. MASLD is characterized by fat accumulation in the liver that is not related to significant alcohol consumption. This condition can progress from simple steatosis to its more advanced stage known as metabolic dysfunction-associated steatohepatitis or MASH. Early-stage interventions are essential for managing MASLD, which is primarily marked by excessive fat in the liver. The pathogenesis of MASLD is complex, involving genetic predisposition, environmental factors, and metabolic abnormalities such as insulin resistance and dysregulation of lipid metabolism. Recent research has also highlighted the role of gut microbiota dysbiosis in the progression of MASLD, indicating that gut-derived metabolites can significantly impact liver inflammation. Health functional foods are emerging as potential therapeutic candidates for preventing MASLD, supported by advanced network pharmacology and multi-omics approaches. Specific food components that may enhance liver health have been identified, detailing their mechanisms of action. Among these, 8-prenylgenistein, a derivative of genistein found in fermented soybeans, shows promise as a potent AMPK activator, improving pharmacokinetics and reducing hepatic steatosis. Additionally, RNA splicing plays a critical role in liver function, with dysregulation potentially leading to issues in liver disease progression. Understanding alternative splicing is vital for enhancing proteomic diversity and addressing liver health. A new minimally invasive procedure is changing the game for those suffering from Achilles tendinopathy. Dr. Vulcano, Chief of Orthopedic Surgery at Mount Sinai Medical Center, has led a groundbreaking study on this innovative technique known as percutaneous Zadek osteotomy, or ZO. The procedure involves making 2 small incisions in the heel to remove a 10mm wedge of bone, which optimizes tendon biomechanics. Patients undergoing this procedure can experience relief within 6 weeks, a significant improvement compared to traditional open surgery that can take up to a year for full recovery. This minimally invasive approach not only reduces post-operative pain, but also allows athletes to return to their workouts within weeks. Dr. Vulcano's study highlights high patient satisfaction and low complication rates associated with this new surgical technique. Analyzing patient-reported outcomes with a minimum follow-up of 2 years, the study confirms the effectiveness and safety of the procedure. While first-line treatments for Achilles tendinopathy often include conservative therapies, over 80% of patients do not respond to these methods. In contrast, the percutaneous Zadek osteotomy has proven to be a more effective solution for those who fail non-operative treatments. Dr. Vulcano points out that many patients suffering from Achilles tendinopathy are non-athletic women in their 40s and men in their 60s. Mount Sinai Medical Center, established in 1949, is the largest independent teaching hospital in South Florida, recognized for its comprehensive care across various medical specialties. Dr. Vulcano emphasizes the significant benefits of this procedure for athletes, enabling them to resume their training much sooner than with traditional methods. Overall, the percutaneous Zadek osteotomy represents a significant advancement in the treatment of Achilles tendinopathy, offering faster recovery and improved patient outcomes. A recent study from the University of Western Australia has uncovered significant connections between abdominal fat and chronic pain in a large cohort. The research analyzed health data from over 32,000 participants in the UK Biobank, averaging 55 years of age, to explore this relationship. Participants underwent MRI scans to measure both visceral and subcutaneous adipose tissue, while also reporting chronic pain lasting more than 3 months in areas such as the neck, back, hip, and knee. The findings indicate that women are more likely to report chronic pain associated with visceral fat, with odds ratios showing they are twice as likely to experience pain related to visceral adipose tissue and 60% more likely for subcutaneous fat compared to men. Conversely, men displayed lower odds ratios, suggesting a gender disparity in how abdominal fat distribution influences pain perception. This study builds upon existing research that has established a link between obesity and musculoskeletal pain emphasizing the specific role of visceral fat in chronic pain, particularly among women. Furthermore, a dose-response relationship was identified between the number of chronic pain sites and levels of both visceral and subcutaneous fat, as well as overall body weight. Researchers propose that targeting abdominal fat reduction could be an effective strategy for managing chronic pain, especially for those experiencing widespread discomfort. They also noted that excess abdominal fat may trigger inflammation, potentially influencing pain perception and the nervous system. However, it is important to recognize that the observational nature of the study cannot definitively establish causation, and limitations exist, such as a small sample size for repeat MR1 scans and the absence of pain severity assessments. The stronger associations observed in women may stem from differences in fat distribution and hormonal factors affecting pain experiences. Additionally, the study highlights the broader health implications of excess abdominal fat, which is linked to increased risks of metabolic disorders like diabetes and heart disease. With rising obesity rates in Britain, where 2/3 of adults are now classified as overweight, these findings underscore the urgent need for effective weight management strategies. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.